Good morning and welcome to the second series for the International Plant Breeding Seminar. Thank you so much for your attendance. And thank you, Paul, for volunteering to and accepting the challenge to be the first one to open up this series. Paul Chavarriaga, Dr. Paul Chavarriaga got his bachelor at the Universidad del Valle in Colombia. Then he got a Master's of Arts at Washington University and a Master's of Science at the University of Georgia. And finally a PhD at the Universidad del Valle. Dr. Paul Chavarriaga works at CIA, the International Center for Tropical Agriculture in Colombia, and he has worked since he started back in 1998 in genetic transformation and gene editing of tropical plants. Thank you so much, Paul, for your accepting this invitation. And I'm going to stop sharing here, and then you can start sharing your screen. Thank you so much.
Some of the figures are showing some cocoa, cacao, cassava, rice, beans, among others. So let me start with the real presentation. First thing I have to note is that CIAT, I would call CIAT from now on because it's easier for me than pronounce the whole name. So CIAT is not the only center that does use gene editing and biotechnological tools for breeding crops. Within the CGR, which is becoming today the 1CG, there are at least 7, I believe, centers that use gene editing technology for crop improvement, trying to, of course, obtain the same objectives, which is producing resilient crops, which are, can stand climate change challenges, trying to maintain the diversity in terms of nutrition, and of course, supporting sustainable livelihoods. The question, I think, is one of the questions is why are we using gene editing? And there are many answers for this. I would try to address some of those specifically from the technologies point of view and regulation as well. As you can tell from this information, there are over 50 plant species for which gene editing technology has been applied. And it includes clonally propagated crops, which are the difficult one, the ones that have very long, let's say, life cycles, sometimes very high heterozygosity, like in the case of cassava, also cocoa. And so it's very challenging, but we know it has been applied in those and in some other crops like monocots, in wheat, especially maize and so on. The clonal crops, for you to work with them, especially for cassava, you do have to preserve the genotype. So segregation is kind of difficult. It sometimes is not appropriate. You don't want to segregate your genotype, tell genes apart, you shouldn't do it. And so gene editing may help you to maintain that particular characteristic. And it does it may help to speed up breeding depending, of course, upon the trait. Not always you can do that with gene editing, but there are several examples which I'm going to bring you about on this presentation, where you can actually speed up the production of new materials by using gene editing. It serves several purposes. We can prove concepts, you can validate gene function if you want. You can even target traits, quantitative traits. After all, quantitative traits are made up of single genes. So you can, when one of those genes is contributing a very high, let's say, percentage to the characteristic, then you can target that particularly. You can, of course, understand much better gene expression, the effect of shutting down or enhancing the expression of genes. All those characteristics make gene editing a very special tool to understand sometimes difficult crops like the clonals. And one of the most important for me is that it does recreate variability, meaning that you can actually use, although allelic variation that you can find in germplasm banks, which is often untapped and very difficult to incorporate because the breeding process takes much longer. So, and I will bring you one of those examples also with cassava. Okay, let's move to the next one. The other answer to the question of why is the regulatory framework. And this is a map that is being taken from colleagues, a paper, a publication from colleagues from North Carolina NCSU, I'm sorry, and sponsored by the Inter-American Development Bank. This particular publication tells about the situation, regulatory situation in Latin America, and but also provides this nice map, very comprehensive, in terms of what is going on in terms of regulation for gene editing. So you can tell that red countries are the ones that do have specific genome editing rules, and in which most of those countries, these products will not be considered as GMO, of course, depending on the final product. Those countries in blue is they are still discussing them. There's some exceptions and special situations. For instance, in Ecuador, in Peru is completely forbidden. And the green ones are the countries that consider gene editing as a GMO technology. So to me, it is important to have it clear because we at CIAT, we do not work only for Colombia. We work for the whole world. What we produce here, what we make here, has to be taken somewhere else in the world. So it's important for us to know who is willing and who can actually accept deployed, use materials that come from gene editing. So that's the actual situation, which is encouraging, especially in the American continent. In Colombia, we do have, of course, a regulation. Since 2018, the Instituto Colombiano Agropecuario ICA made a resolution that established how the procedures for you to ask them if your gene-edited product is considered GMO or not. In Colombia, like in Brazil, in Argentina, in the US and Canada, just to mention some, the final product is what counts. So if you don't have any transgene material inserted in the final product, it may not be considered transgenic. It may be deregulated as a conventional cultivar. That exactly was what happened with waxy corn and rice very recently. One of those, this rice is the one that I'm going to show you later also. So in Colombia, again, the framework, the regulatory framework is ready and is, I would say, very positive because it does encourage institutions like this, which is a public institution. CIAT is a public institution, international though, and but also public institutions within the country and small enterprises and so on to use the technology for their own purposes. I'm sorry if this following explanation is too basic, but I think it's necessary. It's just to remind you who is who in breeding. Of course, most of you may be breeders. I'm not, but I try to play that role sometimes from the molecular part of the breeding. And this is what conventional breeding is about. You basically make crosses. You can use mutagenesis, protoplasm, some other technologies to end up with a variety that is a new variety. It does not contain any transgenes, and therefore it cannot be classified as a transgenic, but it does have, it's been modified genetically because it's not the same variety as the one we started with. So there's modification, but it's not by transgenesis. By transgenesis, then you have the chances of incorporating new genetic material, different genetic material that may come from, let's say, wild relatives. In that case, it's called cis or intergenesis, or transgenesis when it comes from very unrelated organisms like bacteria, for instance. So you end up having a variety, a new variety that's called transgenic because it does carry in its genome a transgenes, essentially. And the transgene is represented here by the yellow star. In the case of gene editing, you end up with a product that does not contain transgenes. Of course, you may introduce transgenes using gene editing, yes, but those are going to be considered transgenic. But let's say if you temporarily, momentarily use transgenesis to produce gene editing, and then you get rid of the transgene by simple segregation, the final product is not going to have it. And that makes them to be classified as non-GMO in several countries. But of course, it depends very much on the decisions of each country and on a case-by-case basis, of course. So we are working currently with mainly the SD1 type of gene editing products, which are those that are where we create mutations by spontaneous repair of the breaks that we produce using the technology. I will explain that next. So again, you can produce this type of mutants, SDN2 and the SDN3. All have differences. Again, the number 3 is probably going to be considered as transgenic in most countries, but the SDN1 and SDN2 may not be. So for the time being, and for the purpose of this presentation, we are concentrating mainly, but for the time being, in SDN1 type of products for gene editing, where you end up having no transgenes inserted in your final product, although you may have had a transgene transitory variety in the past. We do use CRISPR-Cas9, which is the enzyme. There are many more, of course, and this is a field that is evolving constantly, very quickly, very rapidly. But the one we're using right now is Cas9 to create this type of mutations again, where you have produce insertions, deletions, substitutions, and so on. And the objective is, for instance, gene validation or gene discovery, new allele discovery, to produce new varieties, breeding lines. Again, concentrated focus mainly on SDN products, SDN1 products. Now, getting into matter here. This is a summary, a very visual summary of our platform, the gene editing platform. We do work with four crops: cocoa, cassava, rice, and beans, in several traits. I will not going to explain all of them. There's no time for that, but I will touch on some specific examples. But I have to mention that in rice, which is let's say our model, our model species, because it goes very fast. You can segregate. You can have three cycles within a year, 3 or 4, depending upon the variety. So with rice, we can do much more and much faster, and we can actually progress very fast with rice than if you compare rice with cassava and cocoa. Of course, not a good comparison. With beans, I'll explain that later. But in rice, we had worked with male sterility, targeting some genes. We are working with grain number. This is the example that I will explain. We do have waxed rice already. It's not in the field yet, but it's stored somewhere. We have kind of slowed down with this because there's a lot of work to do. We are working on cadmium and arsenic uptake, trying to target some genes that are responsible for absorption of these heavy metals. This is a problem in some areas in Latin America and in Asia as well. And we have validated genes that are involved in rice hoja blanca virus. This is a work that has been done in collaboration with our partner Matthias Lorieux from IRD right now. But and so let me continue on these cases. I'll bring you some specific examples, and let's start with rice. The first trait that we are targeting, one of the most interesting for me at least, is the grain number. Grain number in this case, it's been demonstrated, and those are the publications, that it may be that trait may be conferred by one single gene. It's called the cytokinin oxidase dehydrogenase. This is an enzyme that, its role is basically oxidating BAP cytokinins. And when it happens, when the cytokinin gets oxidated, it stops working. So let's say the effect of producing cell division, differentiation, and so on stops when the cytokinin gets oxidated by this particular enzyme. So it's been demonstrated that mutants, conventional mutants, that do not produce that cytokinin oxidase do produce more grains. And the literature is available. This is just one example. And this is not recent. This has been published in Science in 2005. So we decided to target this specific gene in one cultivar, a Colombian cultivar, rice cultivar, which is called Corpoica llanura 11. This is I think is an important one because it is adapted to acid soils from the eastern plains of Colombia, where there is a major rice growing area in Colombia. There are at least 2 or 3 of them, but this is one of the big ones. But the problem is that soils are very acid there. Aluminum in the soil is very high. Then this particular variety has been developed to be adapted to that particular, let's say, landscape. It's very vigorous. It's lodge resistant. Its earliness is also a trait. It's a good cooking quality, and the best one is that it has resistance to pyrrhicularia, which is very tough. That area particularly is a very hot spot for that disease, and it breaks any known resistant genes so far that has been tested that is being broken by this pyrrhicularia. So that one has a very interesting trait, though. So we decided to target the gene there to knock it out. And this is the first field trial. By the way, I have to mention, at CIAL, we have a special permit from the Colombian government, which allow us to work on gene editing here and take our plant material right to the field, a confined field trial, biosafety confined field trial, where we can very quickly measure, of course, the trait and decide if the line is useful or not. We do not have to go and ask for permit to do this, but this is like straightforward from the lab to the field, which is really an advantage because it does help us to speed up tremendously the process of breeding using gene editing and transgenesis as well, of course. So this is the first confined field trial. You can see there's a protection, anti-bird. There's a barrier in the back. You may not see it. To protect against for pollen movement and so on. And the results from those, the initial result, this is single plant results. So please don't take this as a statistically significant. No, not yet. But we are working on it right now, and you will see it. For me, the interesting thing is that what we saw in the greenhouses at the single plant level for the field grain per plant, number of field grain per plants, total grains per plant, number of panicles, it was somehow reproduced in the field. Again, single plant measurements, so gotta be careful with the statistical interpretation. But this purpose, the purpose of this field trial was mainly to increase seed to do the second yield field trial. This is still a confined field trial. You can see the picture. It's very recent. It was taken 1 week ago or so, no, 2 weeks ago. And this is part of the team that is the responsible. I'm not the responsible, but well, I am, yes, but these are the guys and the girl who are working on this particular trait, grain number. So it is already planted, and what we are looking for is within a couple of months, measure yield and do have statistical data, statistically significant data to declare if those lines are going to be useful or not. With the hope that we will apply for deregulation in Colombia, because and that's going to be the second time actually we do that. But that's going to be the first time that I think any institution from the CG, no, not from the CG system, but in the Big South, is going to have hopefully a deregulated variety, local variety in this case. But the first time already happened, and this is it. This is a picture of the field trials, confined field trial here at CIAD of a rice IR64 variety and CRN sub-1 variety that were deregulated in Colombia as well. That was exactly the very first time. It was like a year ago, 2020, I believe. Yeah. That this rice was declared as a conventional cultivar. So this is a product that was a multi-institution, multi-country effort, big effort, which is actually led by Heinrich Heine University in Düsseldorf, supported by a grant of Bill and Melinda Gates granted to these guys. But in this case, another center like IRRI, another CG center participated in this development. Here we are, University of Missouri, University of Florida, and IRD in France. So this is already out there in the field. Right now it's not planted, of course, but it's been, we have measured several traits there, and I think this is the third or fourth field trial that we have done. The trait is important, is Xanthomonas resistance. Xanthomonas is not present in America so far, but remember the story with banana, FDR4, Fusarium, the tropical race 4, it was never here, it was not in America. Now it is in, and it is in America, and we don't have any sources of resistance, so we are rushing to try to find them quickly and try to incorporate them into banana, so it doesn't become a problem. So this is kind of a preventive kind of breeding. I call it preventive breeding, because you do have very good sources of resistance for a disease that is not yet here, but it may come sooner than later. Let's switch to cocoa. Cocoa, we are trying to reduce the intake of cadmium, very much as I mentioned at the beginning with rice. We are trying to reduce the cadmium intake import through the roots by cocoa plants. Cocoa plants are Native Americans, Latin America, essentially from the Andes, from the Amazon as well. And especially in the Andes, these plants somehow evolved to grow on soils that are rich in heavy metals, one of them cadmium. So the plant knows how to store it, and it goes, it uptakes the cadmium, it takes it gets where in the fruit, and that's what we eat. So today, the international regulatory regulations in general prohibit the commercialization and trade of cocoa products derived from cocoa, chocolate in this case, that contains above certain levels of cadmium. And that's, we, Colombia, do produce very good quality cocoa. We export to the US, to Europe, but we know that there might be a little more cadmium that we want in them. So we are targeting genes, the similar genes that exist in rice, targeting them in the cocoa to see if we can switch it off. This is a research that is being supported by USDA and food, and I think FAST is Food and Agricultural Services, I believe. And the Compañía Nacional de Chocolates, which is the national chocolate company in Colombia, private institution. So this is a dual public-private effort. And of course, our input in the alliance. What we did was, again, look at the literature, find those genes, corroborate that those genes are in the cocoa genomes. We have sequenced around 15, I think, genomes of cocoa. So it is, well, it's simple to get to those genes and find the targets there. So the targets were corroborated in at least 5 Colombian cultivars, which we are working with. We started with a bigger panel, like 10 different cultivars. But when you do gene editing, not only is important the genome. I mean, it's good to have the genome. Yeah, you do have to have the genome. Otherwise, you are going, blind. But when the most important thing, I believe, is the tissue culture system. If you do not have the appropriate tissue culture system, it's very difficult to work with gene editing. So we have developed the tissue culture system. At this today, I can tell you that this is the real picture taken today, 1 day ago, of what we have so far, and what we have achieved is these are some of the first transgenic and/or gene edited putatively cocoa embryos. These are somatic embryos. They have producing beautiful roots, and we expect them transfer them to greenhouses within, I would say, before the end of this year. Remember, this is a very tough crop. Cocoa is not easy to transform. Cocoa is not easy to work in vitro and so on. But we are doing it, and we are doing it again with Colombian cultivars, because of course the private company wants and the USDA, they do want to use real cultivars that are useful for growers. Where are we today? I think we are at this stage, more or less. This is a publication from somewhere else, but this is nice because it is indicating that we are right here. We have to go through hardening these plants, transferring them to greenhouses to do the hydroponics for cadmium absorption testing. This is already set up here at the institution at CIAT. We know how to do that. Then we'll take the best ones to the field, produce flowers and fruits so we can get rid of the transgenes. Remember, this is still transgenic, but here's the step where we can get rid of them and hopefully produce lines that are transgene free for further screening in the field and for deregulation as well. We do have to have permission for deregulation. I mean, we have to ask for deregulation for Colombia if we get some of these lines working very well, and we can demonstrate that they are absorbing less cadmium. So this is a long-term project. We already finished, I think, the first 3-year phase. There is a second phase, which is supposed to be 3 more years, in which both institutions, the USDA and the private company, Compañía Nacional de Chocolate, are going to join efforts to support this. Let's switch to beans, common bean, Phaseolus vulgaris. One of the major problems that we are targeting is nutritional quality, meaning that we want to get rid of those complex sugars that produce that gives you digestion problems, not only for humans, but also for animals. And there are several genes, but we are targeting at least 2 of them, which produce very complex sugar, very difficult to digest in the human tract or the animal tract as well. So for that, we have to develop this transgenic system, or the gene editing system, of course. And we have been partially successful, I would say. We are using agrobacterium to produce these plants that you see here. Were tested. I can declare them transgenic because we saw the signals from the transgenes inserted there, but we weren't able to transfer those, to segregate them into the progeny. We didn't see any progeny inheriting those transgenes. So what this says to me is basically that they were chimeric, and that chimerism avoided reaching the germline where those transgenes were going to be transferred to the next generation. So you can tell here that they may do have the transgene, but it's very weak, indicating that there's not enough transgene in there to be transmitted to the next generation. So this is to say essentially that yes, we can do transgenics, but it's not really, it needs a lot of improvement. So we got in contact with Dr. Josias, on the left, who works very close by with Dr. Francisco Aragao, the famous person who developed the first transgenic beans in Brazil, I think in America, I think in the world. Those were developed by shooting, by biolistics, which is inserting pieces of DNA into the beans and regenerating plants, and then segregated out those pieces that you don't require, but just retaining the pieces, the DNA, the sequences that are required for that specific trait. I don't probably need to remind you that those beans are planted heavily in big extension. This is a real picture of transgenic beans planted in Brazil, and it is already commercialized. Okay, so what we did with them is, okay, I asked them, okay, we need to use your technology. Can you provide the plasmid that you use? And they did. Dr. Josias, he sent us the plasmid, and we're working with it actually. We're using the biolistics to insert this and other genes, of course, for gene editing into common bean cultivars that we selected here. So far, this is the result, still ongoing, very little by little. difficult. It's very difficult. Beans is one of the most difficult ones. It has been one of the most difficult ones for genetic transformation, and I bet it's going to be for gene editing as well. So we have been able to produce some plants with resistance to the herbicide, which is described right here. This is already, I mean, the selection and all the process is already published, so don't worry, it is available somewhere else. But we are using it as a model initially, so we can actually use the system, the selection system, to produce transgenic edited plants and then get rid of the transgenes again, as in the case of rice. So you can end up having a edited non-transgenic plant. This is ongoing. Cassava, the last but not the least. As I mentioned before, we are working heavily on cassava using gene editing. We have already, with support of Ingredient, major starch producer in the world, we got some funding to produce a waxy cassava. So what we did is essentially was to demonstrate that you can produce waxy cassava targeting different areas of the granule-bound synthase, starch synthase gene that has been already mutated naturally in cassava to produce waxy cassava. So we somehow kind of recreated those mutations and created new ones. Actually, we ended up producing more than 40 or 50 different lines, but selected 5 of those that perform fairly well under this field trial that you're looking at. This is a picture of the harvesting of waxy cassava, and you can see this is the waxy. When you test with iodine, the waxy cassava will give you this color, which is brownish, and the non-waxy ones are going to give you this dark purple color. We're working in several traits besides waxy. We are working on herbicide tolerance as a means to select for gene-edited DNA-free plants. We are working on haploid induction, targeting a gene which has a funny name, which is called not like that. It's a phospholipase, actually. We have produced those plants. These are those plants. These are the plants that are already in the field, and we are making hundreds of crosses because this particular gene targets pollen development. And believe me, those flowers, male flowers, barely contain some pollen. So we have to do many, many, many crosses to get the induction of haploids, hopefully. We started recently targeting the sweet 10A and sweet 10B genes in cassava, 2 genes that are involved in susceptibility to Xanthomonas. This Xanthomonas Pathobar manihotis it is present in America. Of course, it is a disease which is relevant in some cassava growing areas here and in Africa as well. So we are targeting these 2 genes to produce a resistant cassava, very much like we did with rice, which I already showed you. And those plants, those waxy plants, are already in the field. We know which ones are monoallelic for the mutation, for the waxy mutation. This is an example. This is line 54. We know only has 1 allele for the mutation that produces waxy. So we are either crossing this with another cassava, which is in this case has 3 different alleles, and produce progeny right here to get rid again of the transgene. Remember, this cassava is transgenic, but is edited. So we have to get rid of the transgene as well. And we are doing also self-pollinations, and some of those are here already producing fruits. I personally have rescued some of them, and many more. So the purpose is to, again, segregate out the transgenes so we can have new waxy lines, breeding lines with different allele combinations, if possible, with different, let's say, performance under real field conditions, but transgene free, so we can apply for deregulation in Colombia as well. That would be the first case, I think, if we get it, the first time that this happens with cassava around the world. I'm very excited, but I know it takes time. But I'm patient enough. So I will continue doing this as long as I can. Last one. The last trait, this is not actually, well, yes, it is a trait. We got together, we have a visitor, Dr. Monroe from University of California in Davis. He came here at the beginning of the pandemics last year, I think. And we showed him around, of course, what we had, the gene banks and so on. He got really impressed. He saw all the potential variability that we had stored in these gene banks. So he came up with a proposal which was fantastic from the point of view of the science and the writing and so on. And so we put that proposal together to explore genetic variability in cassava to, let's say, look for alleles, knockout genes that are related to thermotolerance in cassava. He has been done this before with Arabidopsis, I think. Fantastic piece of work as well, is already published. Beautiful. And he wanted to apply, of course, those principles for mining, allele mining in our gene bank. So essentially what we're going to do here, this is a complex slide, but essentially what it says is that we are going to take more than 400 land races, sequence them, and try to find alleles, knockout alleles that have been produced naturally, of course, during the evolution of the crop, and relate them with functional GWAS for looking for thermotolerant mutations that may be associated with thermotolerance resistance in cassava. And then once we have that information, we of course need to validate those mutations, and that's going to be where the gene editing enters to play its role. And we will do validation of those candidate genes with using CRISPR-Cas9 again. That's it's ongoing now. This has been sponsored by the FFR. It was funded by FFR less than 2 or 3 months ago. We knew it was approved. So we are heavily working on this, producing all the material. It requires, of course, some gardens that we are going to characterize this material in the field. Lots of DNA extraction, sending them to UC Davis for genome sequencing and so on. So I'll later, maybe next year, I'll keep you updated on what's going on with this fantastic project. As conclusions, I would say that in our platform, it somehow is covering more, it's going beyond what we used to call the mandate crops. We don't actually restrict anymore to cassava and rice and bean. No, we have incorporated things like cocoa, for instance. And we are looking into, it depends on the demand. People are asking for tomatoes or lettuce or soybeans or maize. I'm pretty sure we are going to be able to create those gene editing pipelines within this institution to serve the purposes of whoever needs them. Those pipelines can help us to, of course, develop new varieties quickly. Our expertise, it does help to accelerate the inclusion of new crops, not only the ones we already have set up, but new ones. For the time being, we are, as I said, limited to the SDN1 type of mutation, but of course we are looking and we are training people, currently training people on looking for different methods to produce SDN3s or DNA-free methods, which are very important, very, very important for clonal crops. I didn't mention and I didn't show any picture. We do a lot of work on outreaching, especially training, talking about public conversation, presentations, seminars like this one here today. And we do support regulatory framework in the sense that we provide all the technical advice that these people really need in Colombia and some other countries. I've been talking to regulators from the Americas, from all the way from Mexico to Chile in several opportunities. I forgot to mention the IICA has done a very interesting supportive work, the same as IFPRI, in providing materials, but also making those seminars now during pandemics and before the pandemics, online or presential ones. And we will continue that. This time of the pandemics has somehow reduced the ability to, bring people together, but of course, online, we still can do it. But there's nothing, I don't think there's nothing better than getting together, talking. The discussion is much more, let's say, encouraging, I would say. And finally, we want to make CIED, the alliance between Biodiversity and CIED, make that as a node of excellence for gene editing, where we can actually go very straight, very fast from the lab to the field testing, because that last one is absolutely necessary where if you want to prove that the work you are doing is really useful or not. With this, I think I'm going to stop. This is the team, about 13 people that work in the gene editing platforms. Yet to them, they are the responsible, again, they are responsible for this work. I'm just communicating the work. And of course, lots of collaborations, a lot of collaborators from Michael Selvarak, especially, is the one that is in charge of the Phenomics platform where we do this, I'm going back, all these field testing for rice, for cassava. Michael is essential in this one. So if you want to talk to him, let me know. The rest of the people included there, and the funding is coming from different institutions, private, public, the research, roots, tubers, and bananas from the CGI, USDA, National Company of Chocolates, HHU, and the Gates Foundation, Ingredient Flour, the Latin American Fund for Irrigated Rice, that's FLAR, and the most recent UC Davis and FFR. I stop here, Carlos. Thank you very much.
Thank you so much, Paul. Very, very good presentation. I can tell you I miss working with you a lot, man. You have done a great job.
Thank you.
I think it's, I mean, we have in the audience people from here from NCSU that works in and plant transformation. I think I saw Kidonda. I think he probably put good attention to what you and what you presented. Thank you so much. A reminder for the audience: please put your questions either in the Q&A or in the chat. I already have a couple of questions for you, Paul.
Okay.
So the first question comes from Dennis. He says, "A wonderful presentation. Would you please explain what is meant by the term DNA-free methods?"
Well, maybe that's not correct. Maybe it's not, because we have to use DNA always to do gene editing. It's transgene-free methods, which means exactly that you end up having, well, no, that's not correct. Okay, let's rephrase it. DNA-free actually means to methods where you don't use DNA to make gene editing. You can use just the proteins, for instance, the Cas9 protein attached to the guide RNA, which is not DNA, it's RNA. So you can somehow deliver through different methods, transfections, protoplast. We've done that with cassava. It does work, but it's very inefficient. And having to regenerate from protoplast, it's tough, it's difficult, and it does create a lot of somaclonal variation, which you don't want in your final product. One system that is working, I know, is for potatoes. Potatoes are very nice because they allow regeneration from protoplast. So I think it's been done in at least in potato, but also I think in wheat, DNA-free methods. Again, it means you don't use DNA to do the gene editing. But okay, no, I'll stop right there.
Yeah, I think it's we're working here in the case, for example, sweet potatoes. It's very important for those species that you cannot propagate sexually to get rid of the
transgene,
transgenes. Okay.
The foreign genetic material. That's what I'm actually putting both together, because you can do that. You can obtain DNA-free methods to get rid of transgenes. That's true.
So the second question is also a wonderful presentation. The person is asking, is next generation breeding the same as next generation sequencing?
Next generation breeding is the same as next generation sequence. But I think they do have to do each other somehow to do with each other. But No, next generation breeding, I think, relates more to this type of technologies, right? Where you can actually use, for instance, gene editing to go into a particular gene, target a particular sequence within the gene, and do, for instance, a mutation. You can actually create a resistant variety to, let's say, Xanthomonas in rice just by targeting a specific sequence in a promoter of the susceptibility gene. That's a very advanced next generation technology for breeding, but it does require, of course, a next generation for sequencing, because as long the more you can get information on the genomes, the faster you can get them, the better for application of these technologies. Remember, you do have to have information on your genomes, on your cultivar genome. You cannot work for gene editing based on the genomes that are available for some other varieties. Is very risky. So you do have to have a system that provides very fast information of your genomes. So they are somehow interlinked, I believe.
Okay. Another question from the chat is if CRISPR-Cas9 is effective across all plants.
I missed it. Say it again, please.
Is CRISPR-Cas9 method effective across all plants?
CRISPR-Cas9. Well, so far as I mentioned, this is not only for CRISPR-Cas9. There are many more Cas9. Many more Cas proteins today. I don't know how many, probably 10, 12, I don't know. And not all of them have been used. What I can tell is that this is a rapidly evolving, technology that what it makes is people start discovering new Cas9s with different properties and so on. So those have not been tested yet. I'm sure there's a lot of work to do, but editing it goes across crops. In most major crops, 50 of them, it's working. So I would say maybe yes, it's very reliable.
It depends on how effective the delivery system is, isn't it?
That's the point, and that's what I insisted, that not only is having the Cas9 or the right protein and the delivery system, but also the tissue culture system that has to do with how effective is it going to be. There are already methods, at least published, that show that you may not need even tissue culture. Those have, I don't know, we have to test them, like using pollen to do that. But
well, we have here a faculty member, Wushen Liu, who is investigating that. So probably be good to put you in contact with him. And
that'd be good. That'd be good. There's a publication on Nature Biotechnology on one case. I don't remember it was soybean or what, but yes.
Kidong, yeah, I think he's still part of the audience. Another question here, and this is from the Q&A, is asking if you're familiar with a group called Ethos Chocolate, which is a pro-GMO chocolate company, and are you collaborating with them?
The answer is no.
No. Okay.
But I'll be very, very much interested in knowing and getting to know them. Yeah, of course. Because
well, if it's an anonymous attendee, so whoever posted this question, if you can probably post a link to this company, it will be good. So Paul can take it from there. Okay. Another question here in the chat. Thank you for the presentation regarding the alliance between Bioversity and CIAT. Does CIAT now work on any crops that used to work, I imagine, including the crops that other centers work, like SIP and CIMMYT? How is I mean, it looks like you're taking on gene editing basically across different crops, although the priority is on some of the mandate crops. Yes. What is the situation, Paul, in that regard?
Well, today we are moving, the CGIAR is moving towards 1CG, which means a huge institution that actually brings together the work of all of us in different crops. So if we see ourselves as 1CG, yes, we're working in several different crops, but each institution will still maintain certain crops priorities. For instance, we can also share. For instance, we are working together with IRRI. IRRI is the major rice institute. We can working with them, and we can work with them because of course varieties for Asia and Africa are different from the ones in Latin America. So who is going to target those for Latin America? That's probably our role. So I don't think there's going to be like a conflict of interest. No, I don't think we can clear that up very. And we've done it. Okay. As a matter of fact, to present the initiatives that we are submitting for, funding from the 1CG initiatives, which is going to be, for next year. So we know how clearly to separate between each other, and we know what are our targets, and we know where we can collaborate and where we cannot.
Would you say, Paul, that CIAT and the Alliance, considering all the centers at the CG, that you guys are the leaders in the area of gene editing?
We can, that title, we can split it between other institutions because, we're working on it. I think the facility that we have here and the advantage that we have here in Colombia is that we can go, as I said, straight from the lab to the field. So we do produce the plants here, and next month they can be taken to the field for different, and that somehow, of course, gives you a very extreme advantage because you can test them very quickly. But as I said, there are other institutions working heavily on gene editing. IRRI is one of them. CIP is another one. IITA is another one, and I think CIMMYT is doing something with Corteva in maize. So I think we can split that title, but okay, I'm proud to say that we do have an advantage.
How another question here from the chat: how expensive it is to do gene editing?
It is as expensive as it is doing transgenics, probably, because there is a myth. People believe that because this is just a single protein and that you can buy it for, I don't know, less than $100, and then you can go to your home and do gene editing in the garage. That's not true. That's far from reality, because you do need an infrastructure. You need a laboratory. You need genome information. You need specialists. You need tissue culture. You need growth rooms. And that's not cheap, of course. As an average, to say something, you can get a project funded for gene editing, which may cost you at least, I don't know, $300,000 a year or so. Of course, it does include budget for everything, but mostly for your personnel, which is specialized, field testing, the laboratory, and so on. So I would say I don't know, it's not cheap. I cannot say it is cheap, right? But my advice is that it's simple. It doesn't mean it is cheaper, right? And in the end, after the deregulation, where you can, if your product is considered as conventional, then the costs of taking that to farmers or to whoever is going to use it are going to be much lower if you compare with transgenics. That's where the difference may reside. Yeah.
Great, thank you. I think that addresses a question I have below, that is talk about the cost and lab materials and things like that. A couple more questions before we close. One of the questions is how many varieties you can estimate are grown in cocoa or cassava, and what would be a reasonable number to target for gene editing? You cannot target them all.
It's a difficult question because each variety has a, it depends. In the case of cassava, varietal replacement happens very often, and this is somehow undesirable, right? Because stability of the variety out there in the field depends on many different factors. For instance, disease pressure. If resistance is broken, then to a virus, for instance, then that variety is out immediately. It has to be replaced. So I would say that it also depends on what is the final purpose. If it's for a major industrial production, let's say of cassava for starch, industrial starch, then you do have to target, in Asia, for instance, 1 or 2 varieties, which are the most planted ones, right? In Colombia, probably 1, 2, yes. But if it is for, consumption, for farmer consumption, that's a different type of variety, which is not planted heavily, but is extremely important for sustainability, for livelihood sustainability. So it's difficult to answer that question in one phrase.
But definitely, you need to know what will be your target varieties, what the major ones. You cannot address the whole world.
No. It's a case by case basis.
Case by case. Okay. And the last question, I think it's already. I think you answered already, because it's what are the prospects for non-GM based gene editing, and you already talk about this, because it says there's a lot of restrictions. Some countries have restrictions for commercialization of GM crops.
Yeah. That's true, Carlos. Well, thanks for the question. Now, this is one of the issues that we, of course, since we started working on this on gene editing 10 years ago, probably, we started, of course, thinking about, okay, what about perception, regulation, and so on? Because one thing that you don't want is that there is a whole new regulatory framework across countries that separates these gene-edited products from conventional breeding. You don't want that, because that's going to be complicated, and that's going to make it impossible for especially public institutions to end up having such a heavy regulatory framework like for transgenics, because the only final, let's say, what happened after that is that transgenics is basically only few people can do it in the world. And we don't want that. We want everybody else to be able to use the gene technology, the gene editing technology, to produce whatever they think is there for their own agricultural purposes and necessities within the needs within the countries. So, but you have to screen constantly, put your radar and see what's ongoing in the world in terms of. And I know of initiatives of some groups anti-gene editing that are trying to put a framework for gene editing crops. So they are declaring that this is something that has to be regulated pretty much like GMOs, and I don't think that's the case. In the end, Carlos, each country is going to decide and is deciding on either will accept or not gene-edited products, depending upon if they do carry or not transgenes. It's that simple. Once you determine it is not transgenic, then it is automatically a conventional crop, and it has to be treated as a conventional crop. And that's the country's decisions, and you should not interfere with that.
Thank you so much, Paul. I think we are at time for closing, because people have appointments, you have appointments, and I want to thank you so much for the presentation. The recordings will be available soon, and especially thanks to Brandon, Janine, and Anthony for your help. Next week, we're going to have Professor Nigel Maxted from the University of Birmingham in the UK, talking about genetic diversity and maximizing the founded resources for plant breeding. I think Nigel is a recognized authority on genetic diversity for the using breeding programs. So look forward to his presentation, and I look forward to your participation. Thank you so much. And you guys have a nice rest of the week and a good weekend. And thank you, Paul. And
thank you, Carlos, again. Brandon, thank you very much. Bye-bye.
Bye-bye.